Hi everyone, and thank you so much for coming to my talk. My name is Samantha Shablin, and I'm a PhD student at the University of Florida. And today I'm going to be talking about some of my research on the impacts of Batrachochytrium dendrobatidis infection on amphibian stress physiology and behavior. So in recent decades, amphibians have been declining worldwide, and one of the factors implicated in these declines is Batrachochytrium dendrobatidis, which is a fungal pathogen also known as BD that infects the skin of amphibians, which leads to osmotic issues that can result in cardiac failure and ultimately death. However, not all infections end in death, but this doesn't mean that an individual isn't experiencing any effects of disease because sublethal effects can still occur. And most often, when we think of disease or other environmental changes occurring, we believe that the individual subjected to these changes is experiencing some type of physiological stress, and that's what ultimately sparked our interest in examining these relationships between disease and stress physiology. In addition, we were interested in exploring whether disease may have some type of impact on an individual's behavior, specifically their activity levels and exploratory behavior, because both of these are important to a frog's fitness because they're involved in activities such as searching for prey, evaluating territories, or finding protection from predators. And all of these also might suffer some type of consequence from disease. As a proxy for physiological stress, we use corticosterone, also known as CORT, which is a metabolic hormone in the glucocorticoid family. This is because CORT is heavily involved in the stress response of an individual to maintain homeostasis during a stressful event. And these stressful events can be anything from environmental toxins to disease. It's also important to note that previous studies have seen interactions between CORT levels and BD infections in frogs, such as in this figure from a previous study showing the global pandemic lineage, or GPL lineage, leading to increased CORT release rates in midwife toads specifically. So the research questions that I'll be answering today include, number one, how does BD alter amphibian stress physiology? And two, how does BD alter amphibian activity levels and exploratory behavior? In order to look at BD and stress physiology in frogs, I used Cuban tree frogs specifically, and I collected 40 frogs from the University of Florida's campus and split these 40 frogs into two groups, a BD exposed group, which had 25 individuals, and a control group, or a BD negative group, that had 15 individuals. Then I took urinary samples to use for CORT measurements from the frogs pre-exposure to their treatments, followed by inoculation with either BD or water. And then every 10 to 12 days after infection with BD, I would take another urine sample from all of the frogs. And this happened 3 times in total with 3 rounds of BD exposure and 3 rounds of urine samples. For my second question, I constructed these arenas with trees and assayed frog behavior 3 times. Frogs were powdered with this fluorescent pink powder that you can see in these images, so it would be easier to see their movements in 20-minute video recordings, because these videos were taken at night when the frogs are most active and the videos were pretty dark, so the pink powder made it easier to see them in the recordings. And we did 3 assays in total, with the first assay including immediately after capture in the field, and then the next assay 24 hours in the lab to see if there were any differences between a field setting and a lab setting. And then our final assay happened post-exposure to their treatments after the 3rd round of BD infection. And then in each video, we counted the number of trees climbed and then the total time spent moving for activity and exploration. And to put this all into one consistent timeline for CORT and behavior, we first collected frogs from the field, took initial measurements and tested individuals for BD, assayed their behavior in the field, brought the frogs to the lab, took the first urine samples pre-exposure, assayed their behavior before exposure in the lab, infected the frogs, swabbed frogs for BD load, and 10 to 12 days later, and collected their second urine samples, infected frogs again, swabbed frogs for BD load 10 to 12 days later, and collected our 3rd urine samples, infected our frogs for the last time, and then 10 to 12 days later, took our final measurements and urine samples. And then also we conducted our very last behavioral assay that happened post-exposure to their treatment groups. And we did qPCRs and ELISAs to determine our BD loads and our CORT measurements. So on this first graph, we have time point on the x-axis when each quart measurement was taken, and then on the y-axis, we have the quart levels. What we found was that the infected frogs at the last time point had higher quart levels than the control group or the BD negative frogs, which is at this fourth time point. In our next two figures, we have time point at which the behavioral assay was conducted on the x-axis and activity level or number of trees explored on the y-axis. Note that each box denotes both the control and the exposed groups together, just before or after their treatment exposure. And what we found here in the figure to the left is that frogs became more active post-exposure, or the longer they spent in the lab, and then on the right, frogs became less exploratory post-exposure. And so to reiterate our findings, quart levels were associated with BD infection, but only at the final time point. So this really emphasizes the need to take repeated quart measurements over a period of time. Otherwise, you might miss some associations that are happening later on. In addition, treatment did not have an effect on frog activity levels or exploratory behaviors. So BD doesn't seem to be playing a role in these specific behavioral changes. Finally, behavioral changes in frogs did start occurring, but only after some time in the lab. This is also important because from the field to the first pre-exposure assay, there were no differences in their behaviors, but from that pre-exposure assay to the post-exposure assay, there were differences. And this might be because their surroundings are changing, such as they're getting fed more regularly, they don't have to worry about predators, etc. And all of these could be contributing to some associated changes in their behaviors. I'd like to thank all of the members of the Keiser and Longo labs for their support, especially throughout the past year and a half. And thank you to Steve Johnson for all of the frog collection equipment, the Natural Area Teaching Lab at UF for their facilities, and then last but not least, the NSF for their funding. And thanks for listening, and I'm happy to take any questions.